This presentation is a highlight of research conducted in the Shehu Computational Biology Lab at George Mason University. This research is based on the following conference and journal publications. One of the central dogmas of structural biology is that protein sequence to a great extent determines the three-dimensional structure of the protein under native conditions. This shape, also known as the native structure or native conformation, can then be used to imply the protein's function. Therefore, determining the structure of a protein is an important link in expanding our knowledge and understanding of many diseases and will assist in the design of novel drugs. Over the last 3 decades, we have seen rapid growth in protein sequence data. As shown by this chart, discovering the three-dimensional native structure of these corresponding sequences remains challenging. Experimental methods have failed to keep up with the rapid growth of sequence data. The Protein Structure Initiative Project has helped organize and coordinate experimental methods of solving protein structures. However, the gap between known sequences and known structure continues to grow at an exponential rate. Computational methods are needed to supplement experimental methods to help close this gap. Our methods are inspired by robotic motion planning techniques. Let's quickly review this class of algorithms and how they can be applied to study protein motions.
Path planning algorithms find a path from a start configuration to a goal configuration while avoiding obstacles, shown here in blue. In protein systems, higher energy states will act as our obstacles. This picture illustrates a very simplified problem with 2 degrees of freedom. The high energy areas are shown in red. This video illustrates the process of navigating an energetically feasible path between the start and goal states using a path planning algorithm known as RRT. The algorithm works as follows. A new point is sampled at random. The nearest node in the tree is then located, and an attempt is made to extend the node towards the sample. If the new location is lower than energy, the move is accepted. If the move is higher than energy, then the metropolis criterion is used to determine if it is accepted or rejected. Our work extends this algorithm to work on protein systems. Since even coarse-grained protein representations consist of hundreds of degrees of freedom, randomly sampling conformations in order to extend the search tree is not practical.
To locate minima in the protein energy surface, a tree is grown in conformational space starting at an extended conformation. Nodes are expanded through short metropolis Monte Carlo runs that utilize molecular fragment replacement for constructing the move set. Tree growth is biased towards low energy and geometrically distinct areas of conformational space. This is accomplished by 2 projection coordinates that are utilized. One is based on energy, shown here on the left. The other is based on a geometric hash that groups together conformations with similar geometric features. Our work investigates different heuristics on these 2 steering mechanisms to find the ones that yield the lowest energy ensemble of conformations that are geometrically distinct. A node is selected for expansion by first selecting an energy level to expand, shown here in green. Biasing schemes are used to steer the tree towards lower energy levels. All of the nodes within each energy level are projected into our geometric grid. A cell is then selected from the grid, shown here in green. A weighting scheme is used to prevent the same cell from being selected too often and to encourage growth in undersampled areas of conformational space. One goal of these heuristics is to sample a more diverse collection of low-energy conformations. To evaluate the different schemes, we first remove higher energy conformations from the tree. Next, we perform clustering to remove proteins which are very geometrically similar to one another. To evaluate the effectiveness of different heuristics to sample a more diverse ensemble of low-energy conformations, we inspect the reduction achieved by the clustering algorithm. Lower reduction rates indicate a more diverse ensemble. This chart shows that an energy bias based on the Gaussian distribution produces a more diverse ensemble compared to a grittier method known as quad. The softer energy bias maintains the ability to sample conformations close to the native state, as shown on this bar graph. We conclude that our enhanced sampling strategy improves the coverage of conformational space while maintaining the ability to sample the lowest energy conformations.
Many proteins tune their biological function by transitioning between different 3-dimensional shapes. This slide shows 3 states of commodulin. Each of these protein structures is comprised of the same amino acid sequence and represents a system with several thermodynamically stable states. This protein can therefore act as a molecular machine, changing its three-dimensional shape to carry out different biological functions.
Modeling the transitions between these states is challenging due to the large number of degrees of freedom and the large amplitude of the structural changes involved. Our tree-based strategy can be applied to this problem, defining both a start and goal state, and biasing the growth of the tree to connect the two states together. Let's review how nodes.
The process of expanding a node employs the molecular fragment replacement technique. This technique greatly simplifies the search space by utilizing a database of backbone angle vectors custom built for our protein system. A new candidate conformation is formed by performing a single fragment replacement on the parent. The energetic credibility of each candidate is evaluated using a potential energy function and the Metropolis criterion. Our method creates M of these candidates. The RMSD between each candidate and the goal structure is then calculated. The candidate that moves us closest to the goal structure is retained and inserted into the tree. A projection layer is employed to bias the growth of the tree. Each conformation is mapped into a one-dimensional grid based on its RMSD to the goal state. Selecting a node for expansion is a two-step process. First, a bin is selected, and then a conformation from within the bin is chosen. We investigate different methods for selecting the bins. Some are greedy and pick the nodes closest to the goal more frequently. A slightly softer bias towards the goal has been shown to yield a good balance of exploration, shown in the breadth of the search tree, and depth, shown as the proximity to the goal state. This chart shows some results of employing our framework on a few representative protein systems. The different colored bars represent the different biasing techniques used to guide the growth of the search tree. The results show the RMSD distance from the closest node in our tree to the target structure. This video shows the pathway discovered by our framework for a chymodulin, a protein with 144 amino acids. This video shows the pathway discovered by our framework moving from the closed to open state for adenylate kinase, a protein with 214 amino acids.
This research is made possible in part by funding from the National Science Foundation.